Bonjour, mes amis. Hello, my name is Dr. Krista Kostroman, and I'm Chief Science Officer here at the DNA Company. And I'm delighted to be here with you all tonight to be discussing how lifestyle choices, or our day-to-day living, or day-to-day health choices, impact our genetic expression by way of epigenetics, a very fun, fancy word with a lot of fun and fancy things tucked into it. And I'm very, very grateful this evening that we have our in-house clinician, Dr. Lara Varden, who is a PhD doctor, who some of her PhD did cover epigenetics and clinical applications, health applications, and all that glorious fun stuff. So I'm grateful that I have an expert on the subject matter here to be discussing. So we want to make sure that this session is for you, supports you, really empowers your health journey, and shares with you what you want to know about your health, but specific to tonight, what lifestyle things you're doing that impact your epigenetics, namely impact how your DNA is expressing. If I'm using a lot of big fancy $20 words there, the answer is yes. There's going to be a bit of terminology that we're going to go over tonight. And so how we want to work through today, we're going to start off with a little bit of kind of discussing what epigenetics is and is not. There's a lot out there in popular science that is, half true, needs a little bit more context. So we're going to start off with just a basis of what epigenetics is, what it means to your health. And then the really fun and important thing, at least for me as a clinician, I always care about the so what of science. We're going to talk about what this means to your healthcare. What does this mean to your personal healthcare plan, and how you can implement new technologies that look at epigenetics, measurement thereof, within your healthcare plan. Yeah, and again, we really want to make sure that today's session is for you and about you. So in a second, I'm going to introduce Dr. Lara. And as I'm speaking now, as she is speaking and introducing herself, I really, really encourage you to fill out that poll that we put, as we were waiting, as that glorious music was playing. Please do fill out that poll, because that's going to educate us why you're here, what you're caring about, and make sure that we really tailor to that in today's session. And for those that join some of our sessions, if you're someone that's going to click other, I really encourage you to put that other into the webinar chat. Also, for those who already know this, I'm still going to offer the lay of the land to those who may not. Dr. Lara and I will probably talk for about 40 minutes about the glory that is epigenetics, and then we'll close off. The last 40 minutes are going to be dedicated to question and answer. So if you have questions and answers, please do put them into the Q&A chat. We tend to keep that open until we get to a point where we don't think we're going to fill up, or we're going to exceed, 40 minutes. So please, I really encourage you, right now, if you have any burning questions, toss them in. If you have any questions as the session goes forward, toss them in right away, because we want to make sure we get you in the queue and we're not cutting you off. So with no further ado, as you're entering in questions, as you're filling out surveys, I would love to welcome the most wonderful Dr. Lara. And Lara, I wonder if you can introduce yourself a little bit, talk to us a little bit at a high level, what your PhD was about. Bring it down to earth for us humans down here.
Well, hello, and thank you everyone for joining us this evening, or afternoon or morning, depending on where you are in the world. We appreciate you taking your time out to learn with us, to discuss these wonderful, glorious things, as Dr. Kostroman was talking about us, and tonight being epigenetics. So as she said, I am a PhD, so I did most of my work in the lab, on the bench, doing all that wonderful research that usually doesn't get seen or put into practice for 10, 20 years. And that's actually what brought me to doing what I'm doing today, because I didn't want to wait 20 years to see all that wonderful work. I wanted to have that more immediate contact with clients and teaching them about things that I know, that I've experienced, that I have learned and continue to learn along the way. And I learn every day from every one of my clients. And I'm so grateful for each and every one of you out there, and all those who I will be working with in the future. So for my PhD, now this is kind of a mouthful. I actually got my PhD in interdisciplinary biosciences and biotechnology, specializing in molecular genetics, cellular biology, neuroscience, biomechanical, biomedical engineering, a bunch of other things. So I danced around between many labs doing lots of fun things. So really, what does this mean for you, and how does this qualify me to talk tonight? Well, a lot of what I talked about, or a lot of what I researched and published on, was about the genes, the genetic expression in a disease state, and then regenerative strategies to manipulate through epigenetics these expressions of the genes so that way they would be in a better healing, more normal, natural state. So, I mean, if any of you nerds out there ever want to Google Scholar, type in my name, you can look at some of my publishings, read my dissertation, collecting dust on the bookshelf. So,
Oh, not at all, not at all. Fellow nerds, check out that stuff. It is all to the end of learning and propagating.
yes.
So before we dive in, because I think my next question for you really pointedly is going to be, what exactly is epigenetics? Let's offer it a real human definition. But before that, I wonder if the powers that be can display for me the results of our survey. Ah, there they are. I appreciate the powers that be. Thank you. Ooh, interesting. Okay, so we asked, the question was, what aspect of epigenetics most interests you? The first option we had, and this is tied for number one, the first option was impact on chronic disease and prevention thereof. That was tied at, so 40%. Most of the people here, coupled with the next one is role in longevity and aging. So there's a happy split between the idea of how do I live forever? And then in doing so, how do I make sure I feel great and I'm not subject to all of these long-standing diseases that maybe take away the quality of life and the point of longevity? There are approximately 11% of you that are curious about epigenetics because they have an influence on physical health, or fitness, I guess thereof, gym folk. And then lastly, there's others. So there's about 5% of you who have other, and I would value and really appreciate it if you toss the other in the chat, because we want to make sure we include the other within these conversations. So thank you for answering that. And we'll make sure as we progress, I think in particular in more that clinical application, how do you leverage this into your health? We'll speak to both the chronic disease side of things and then the longevity side of things, because I think the testing of this and the leveraging of this knowledge lends really well to both of those outcomes. So, Dr. Lara, wrap to me a little bit. Talk to me about what exactly is epigenetics. Can you bring that definition down? Because a lot of the time I think people come up to me and they're like, oh, you know, I've had my DNA tested. I don't really need to know anything else about my DNA. And we know that's not entirely true based on epigenetics. And then there's also people that ask the question of like, hey, now that I know my DNA, I must know everything. It must be everything that is my health. And again, not always the answer. So bring us down to earth. What is epigenetics and why do we care about it?
Okay, so first of all, I'm going to put on my teacher hat. And I do have to say, I actually am a certified teacher. So with that, I really like to get into looking at the words, the etymology, you know, where do these words come from and what do they mean? Because that really gives us a lot of insight on what it is. So let's break it down. Epigenetics, epi being Greek for on or above. Genetics, well, that's relating to our genes and DNA. So what we're looking at is what is around, on, and affecting our genes. Epigenetics is the study of how your behaviors and your environment can cause changes that affect the way your genes work, how they are expressed. Now, I want to be very clear about this, that unlike genetic changes, epigenetics are changes that are reversible. They do not change your DNA sequence, okay? This is really important for you to understand. but they can change how your body reads a DNA sequence. Now, I have this wonderful image that I want to share because I just love it. Can everybody see that?
I can, and God will educate us if no one else can, but I think you're good to go.
So this is a nice little summary when we're talking about epigenetics, okay? That if you look down below, your genes plus your environment and lifestyle equals you. And it's important to keep in mind, like I was saying, you know, as far as DNA doesn't change, epigenetics is reversible. But when we put the two together, you know, like, okay, many of you might already have your DNA 360 reports done, or you've had DNA done, and you're like, okay, well, how does this work? Well, DNA loads the gun. It tells you what your propensities can be, you know, and we've talked about this through several different webinars, but it is the environment, your food, your stress, all of these different things that pulls the trigger, whether you are going to be fulfilling those particular propensities or not. And this is what we do here at the DNA Company, is we help you to decode your DNA, to explain your profiles that you have in the reports that we provide, help you understand, okay, so here's the hand that I'm dealt. Well, okay, here's my gun, my loaded gun. I don't want the trigger pulled. How do we not have the trigger pulled? Or how do we do it in such a way that it is to our benefit?
Yeah.
What is what we do?
An analogy that I heard once around epigenetics was, you know, if you inherit a cookbook and this cookbook has been passed down through the generations, right? You can see the original text of the recipe, you know, add a cup of flour here, do this, do that, use this temperature. But epigenetics is almost the loving notes from your grandmother, from your auntie, from your uncle, from whoever the pastry chef in your family is, kind of very lovingly say, go do this, go do this. Hey, it says a cup, but you might want a smidge more. Right. It's kind of that loving information from both the way you live your life and, to an extent, ancestors as to how you want to be expressing that DNA. Does that make sense? Is that fair, Dr. Laura?
Oh, absolutely. And I'm going to take it a step further. For people, for those biohackers out there talking about longevity, it's like, you know, doing those things, following that recipe in that cookbook, and even adding in those little special things. How can we turn the dial a little more? How can we make it even better? Is the ingredients that we use to be of the utmost, you know, best version. For example, don't just use regular flour. Use organic flour, you know, or, you know, oh, throwing in the chicken. Well, don't use just regular old chicken. Make sure it's organic and free range. Okay. So the ingredients make the difference. I know many of recipes because we've always had our own garden, our own organic garden. I'll tell you what, the taste of the foods, of these vegetables that we harvest from our garden and take and eat immediately, so different than that you buy at the store.
Yeah. And that's such a tempting rabbit hole, quite literally and metaphorically, to do. But I want to make sure we stay on the fun that is science and our little mini lecture that you are offering us. We've all gone back to school. I should have brought my dunce cap. So I guess the next question I have is, how do we go about measuring this epigenetic imprint? And in fairness, I want to maybe you can speak to this first. In fairness, when we talk about epigenetics or things that can modify all those notes in the margin of the recipe, there's many different mechanisms, I believe 4 to be exact, maybe 3 or 4, that enable that sort of modification of the DNA in such that express this, don't express that. So I wonder briefly today if you can talk just quickly maybe even list them off, but really focus on the one in particular that right now we as a science and medical community are leveraging when we're talking about DNA methylation and how we clinically use it.
Okay, and actually, you were right in saying 4.
So
So,
I got nervous and I was like, wait, I can only list 3 in my head right now.
and actually, one of them controls a couple of the others. So the 4 that she is referring to is DNA methylation, telomere length, siRNA, which is short for silencing RNA, and histones. Now I know that's like words. What are these? And this is, yes, a scientific word salad. So really the ones that have been used the most and that are most commonly found in biological aging testing nowadays is DNA methylation, which is the gold standard for measuring biological age nowadays. And I do want to explain the difference between biological age and chronological age. So let's start with chronological age. Chronological age, how many times you've been around the sun every year, your actual birth date age. Your biological age is the age of your cells, of your tissues of the body, because I'm sure that you have seen 2 people say they're both 74 years old. You look at one and yeah, they look their age, or they might even look like they're in their 80s. And then the other person, you'd be like, there's no way you're 74. You've got to be in your 60s. Okay. It is that visualization from the health of their body, their skin, their hair, all of these different things that is affected by epigenetics and through our biological age. Like how young are you?
The question, how long has the calendar said you've been alive compared to how long your body thinks it's been alive?
And how it's functioning. So DNA methylation is the gold standard on how to measure that. The other one that is used often is the telomere length. Now, telomeres are like the end caps, the shoestring, plastic end caps on shoestrings. Okay. Every time a cell divides and creates daughter cells, the DNA actually on these telomeres, they get shorter. They keep getting cut off. Okay. I'm not going to go to the whole thing on how all that works, but just know that they get shorter. So the more replications, the older the cells, okay, the more you have, the shorter that telomere length. And so therefore, that can be a way for us to assess age, biological age. Now, that is not necessarily something that always gets shorter. There's also something called telomerase, and there's different ways to actually make those telomeres longer. And we actually will touch in on some of that just through health recommendations and other things, but we're not going to focus directly on that right now.
Yeah, and I think the net takeaway more of this conversation is telomere length was kind of our first kick at the bucket when it came to how do we assess chronologic age compared to biologic age. And there's correlations, but we found a new technique, namely DNA methylation, that has a better correlation or a stronger predictor of where you're actually at. So it's kind of like going from, oh, I don't do enough TV, but kind of like a standard TV, and then you get like a super high definition 4K TV, right? The fuzzy gives you the point of the movie, but once you get 4K, you can see all the details, you can see all the hairs and the lines and all the things.
Things we want to blur out.
Sometimes if it's not a favorable result.
So the last 2 are not as pertinent, but because I threw it out, I'll just give a brief little thing. For siRNAs, these are, like I said, silencing RNAs, mediate silencing of targeted genes, and these are non-coding. Again, a little more into the weeds than we really have to, go into, but with the histones. Now, when we talk about common epigenetic changes, this is in histone modification modifications. Now, histones are these structural proteins in the cell nucleus, and you can think of it like a thread bobbin that you string your thread around, like the DNA being strung around these histones. And it is with all of these that ends up creating the structure of the chromosomes themselves. And when the DNA is wound around the histones, they can't be read. They cannot make the messenger RNA to make the proteins. So therefore, it is a silencing of them. And with DNA methylation, and that's why those two are connected, where DNA methylation actually affects the histone regulation, is with DNA methylation, it is the methylation, a methyl group, a carbon, 3 hydrogens, attaching to the DNA and making it quiet. And that's where that wonderful, fun cartoon that we had put up first comes into play.
Thank you for that. That might be my new favorite cartoon right now. So I guess let me ask you a question. If I were to get my DNA methylation done, what exactly does it tell me? Can it tell me that this gene is turned on or off, or what exactly is it telling me when we kind of look at DNA methylation right now?
Well, when you get the results of some of the biological aging tests out there, it is not that, oh, this particular part of the gene, this one is silenced in this. What they look at, I mean, yes, they look at that, but they don't explain it that way, because in the different groupings, for say chronic inflammation or diet or, mitochondrial health or something like that, they have a library of hundreds of genes that they're actually looking at. And what they do is they look at the parts of the gene, they're called CPG islands. It's a section of the gene that actually gets the methyl group to make it quiet. Okay. So there's a pattern of these methylated genes. And so we're looking at that pattern and looking at it and testing against the library of thousands of other individuals, based in different backgrounds, socioeconomic, all the whole plethora. So that way it's not just European white men, which unfortunately has been, the group, for so many years in science that's been tested against. So looking at that and then saying, okay, so based on these groups, people who are this age, who have this kind of health, etc., where do you lie? And how do your patterns, how do they match up and determine? So it is not perfect. It is ever evolving. And these are tests that provide wonderful insight and information. It is a piece of the puzzle. It is not the entire puzzle. So, make sure not to put all your eggs in one basket when it comes to these tests, that it really just provides a more enriched picture of you and what you can do to better your health and move toward the goals that you all have beautifully shared in that poll.
Yeah. So how are you then? So now we know DNA methylation testing does not tell us about a specific gene, but more patterns. What are these patterns associated with? So like what health outcomes can we know based on the pattern? So can they tell us everything, or is it specific to we have patterns associated with this lifestyle or diet or whatever outcome it might be?
Yeah, it's more specifically grouped because it is based on research, on the brilliant shoulders of all those scientists, clinicians, everyone who has been, giving their brilliance to the pot we call science. And so we have to look at that and use that information, that published information, a plethora of published research to base this information off of. It's not just some willy-nilly and, oh, this is what we have. No, this is based off the knowledge that we have so far, which is ever changing. So this is something that, it's constantly evolving. So for different topics or areas of concern or of interest, things like I had said, your diet. I know that the diet sections, the genes that are looked at, I believe that there's about 147 genes that are looked at and tested for the DNA methylation pattern, something around there. And it is tested against, well, the diet. Lots of different diets out there, but we know in the blue zones, the people who do live to be over 100, who are very healthy, looking at the Mediterranean diet. And there's also the healthy index, healthy alternative index of food. It's a profile of eating habits that Harvard Medical School actually created. And these are used as the basis of what a healthy diet is. And the methylation patterns, based on that for people who eat like that as compared to those that don't. So other things, like I said, chronic inflammation, mitochondrial health. In some of these, again, they look at a lot of different methylation parts of the genes, and these are not all the same genes in each grouping. Okay. Say there's, a couple hundred genes, in this particular group, and they look at that and say, okay, so for example, with your immune health, okay, they're looking at your neutrophils and lymphocytes, not the number count. This is very different than your blood work. Okay, your blood work actually does a count of how many of these white blood cells you have. With DNA methylation, it looks inside these neutrophils and lymphocytes inside and looks at the DNA in these particular cells, methylation pattern thereof, which can reflect the health of your immune system.
So this, if I'm hearing you from like a clinical perspective, there's an element of this is a snapshot of your body at this moment in time, how it's aging, and can also serve as a measure, a metric over the long term of here's where my body is, here are changes I'm going to make, diet, lifestyle, whatever they might be, and is my body truly registering those changes at a level where it's changing the way it's expressing, changing the way what it's doing, be it improving its immune function or not, should the changes have not clocked so well in the body.
That is correct. And so, but I do have to say, both in blood work and in this, DNA aging or this biological aging testing, yes, it is for that snapshot in time, especially with blood work. Okay, because that is changes much more readily, much more quickly within just, a couple weeks time and even a few months time based on the life of red blood cells. Okay. So with biological aging testing, that takes a little bit longer to actually see the changes. I mean, they have shown some changes within a few months time. But if you really want to see real changes made, it is better to wait about 8 months to 12 months before you do a second test to look at that metric, to see that benchmark. Because, and obviously this works into, if you want to see changes, you have to implement the changes. You have to implement these recommendations and these healthier environmental factors of diet, eating, lifestyle, everything else. Because, if you only do it for a month and then you stop doing it, then you're not going to see much change. Yeah. So,
yeah. And I think especially as a clinician, I find it really valuable feedback in concert with other elements, right? We do not recommend this as your sole and only testing when it comes to health, but it's something really helpful in the sense that it's another means to getting at something that's very intangible, right? A lot of people, for example, will come to us and say, hey, my genes support me doing a vegetarian diet. I'm going to try it out. Let's see how it goes. I feel mostly okay. And, this is another way outside of some of that subjective, how does that diet feel for the body? How is the body responding properly to that diet? because we can see the influence of diet on the genome and the way it is expressing. Is that fair?
Yes, that is fair. You know, I really would like to jump back a little bit when it comes to epigenetic changes, because I want people to realize that the DNA you have is the exact same DNA in every single cell of your body, of the trillions of cells, okay? But yet, in the tissues, oh, I've got skin as compared to heart, as compared to hair, and it's like, but it's all the same DNA.
Yeah.
Well, this is the glory in part of epigenetics and silencing of certain genes that, you know, as an embryo and as those cells are developing, and then we are creating these different cells types and tissues, okay, that all is very important, and certain genes get silenced. So when you turn on and off genes, which is what epigenetics is, okay, it is really important to understand that the proteins that are being expressed, okay, you know, for example, like bone growth, obviously we want bones to grow, the DNA to be expressed, the proteins to be expressed for bone growth in bones, not in muscle tissue, okay? We want to make sure that the understanding that when we have the cells and the tissues that we have, and to make sure that they are regenerating and doing well, that we are giving it the necessary nutrients to do so. This is where the environment comes in, and that within the different tissues of an individual, even in the different cells and everything else, these environmental influences, such as your diet, your exposure to pollutants, those can all impact your epigenome. So wanted to make sure you understand the impact of that, because it really is everything that you do, good and bad, will affect your genetic expression.
Yeah, the epigenome to an extent being a story of how you are expressing in that moment based on what has been happening to you over the past lifetime, past few months, depending on kind of how dramatic those changes might be that you decide to implement against your health. Yeah. I think before we transition into questions and answers, Dr. Lara, is there anything else that any other kind of tidbits or thoughts that you'd like to offer that we haven't covered when it comes to testing DNA methylation before we jump into more the questions and answers, which we're seeing fill up a little bit here?
Actually, yes. I know that there has been wonder about, well, I have my DNA 360 reports, I have my DNA reports. Why would the DNA aging or these biological aging testings be important? First of all, they're not the same, okay? They are two very different tests. Where your DNA 360, your DNA testing, that's the thing that doesn't change, okay? That will remain standard. But the DNA, the biological aging testing, this can, and that, like blood work, like your gut 360, looking at the microbiome, looking at, you know, any kinds of these tests, gives you that metric on which to say, how am I doing? Am I getting worse? Am I getting better? How is this change being done? So when you understand the difference between the two tests, it can help you to better appreciate what they have to offer and how they can complement one another. The other thing that I wanted to mention is for the biological aging testing. Oh, I'm so sorry. I just lost my thought train. Okay, well, we'll get back to that. Sorry. Squirrel.
I adore it. For our audience, both Dr. Laura and I have had a long day, and so thoughts are occasionally escaping our head within this word salad. Oof. Okay, so Dr. Laura, if you remember it as we're jumping through the questions, just kind of let us know, and we can kind of divert a little bit. But I would let's jump into the questions. Let's start supporting people where they're at and what they're curious to know. So Nancy offers us our first question. She says she's interested in the effects of intergenerational epigenetics, both mental and physical health. So I'll lay the stage a little bit and then allow Dr. Lara to jump in. So intergenerational epigenetics, I think the most famous study or the most important or most popular, I suppose, reporting on this was really looking at Irish folk who experienced an incredible famine, the potato famine. There was marked changes of their epigenome, so that which came above in their children, such that it actually changed the way their children's DNA expressed. It was almost this storytelling from one generation to another saying, hey, if famine, then this, and it marked the stress and other such things were seen on their DNA. I think this was one of the, if not the first, one of the bigger studies that kind of said, hey, we need to look at something beyond DNA. So Dr. Laura, any further thoughts, any further insights, especially because I think you've done an incredible job capturing how day-to-day lifestyle kind of within one lifetime, your epigenome can change in response to environment. Can you speak at all to kind of the intergenerational or how that storytelling is propagated?
Absolutely. So remember that epigenetic modifications can be maintained from cell to cell as the cells divide. And this is where, in those cases, they can be inherited through the generations. Because what ends up happening is that when a woman becomes pregnant, but even before that, when we're talking about the eggs, when we're talking about the sperm, remember that these are cells and they are affected by the environment around us, by the nutrients we get or don't get, depending on, the situation, or the toxins that are being, that are affecting the cell and the way it's being read or silenced or anything else to it. So now put the two together and now you have a growing fetus. Well, that growing fetus needs a lot of nutrients and they are going to take it from the mother. Do you have, as a mother, do you have all the nutrients to feed that child? All of the omega-3s and DHA, to help build strong, big brains, to have all of these complexes being growing normally. And it is within the genetics, and as this is growing, that can be affected. That, remember, the epigenetics, it can be maintained from cell to cell as they divide, especially when you've got one cell going into trillions of cells. It's impacted more, and that can carry on through the generations.
I love that answer. Thank you. Thank you. And I think you mentioned mums, right? I think they're starting now, especially to prove how dad's health will also impact that.
Yes.
Yeah. It's on both parents at this point in time. I think just mums were originally explored because obviously. All right. Our next question is going to be coming from Madonna, and I'll supplement a little bit. Diane's also asked us a question, and there's a little bit of parallel between this. I'm not entire parallel, but a little bit. So Madonna's saying that she's made a few bad choices from a lifestyle perspective, poor diet, not exercising, kind of that sort of thing in her past. And she's wondering how much she can reverse her current health issues by changing her choices for the better. She's saying there's nothing currently catastrophic going on, but when she looks at her lab markers, and I'm imagining this isn't necessarily epigenetics, but just general blood work and other such tests, she's seeing a downward trend. Dr. Laura, I'll allow you to answer, and then I want to make sure I bring up a really important study that looks at people who were 50 and had a heart attack. Yeah, lived to be 100. I think it's a really important thing for people to be mindful of, but I'll let you jump in first there with the, can we change? Are we destined to our past behaviors?
Well, the answer to can we change? Absolutely. And are we destined for that downward path? No, not at all. The only way that you become destined is if you decide not to change your current behaviors that put you in that state or toward that state in the first place. So it really is under your control, for the most part, because obviously, location, location, location. If you live near a toxic waste load of things, then I would suggest you to move. But that being said, yes, you can make changes that can get you healthier. What they are in particular and what the best way to go about that, this is where people like us come in, and people who are familiar with your genetics to be able to take a look at specifically where are you weak, where are you strong, what do we have to leverage, and also finding out about the details of your life on what could have been causing those issues, and then rectifying those situations and putting you on the right path. So, I mean, honestly, my thoughts is it's never too late until you're dead.
I'm going to underscore that. I'm going to come back to you in a second with kind of a clarifier from Diane's question, but to Madonna specifically, there's a really beautiful observational study done a little while ago, and it looked at people who had made it to 100. They were 100, as well as they were healthy and happy. So these aren't, 100-year-olds that are wishing their time was coming. These are people that are still joyous in life and still engaged in life and physically active, etc. And they had another qualifier for this population that I thought was interesting. This entire 100-year population also had to have a history of cardiovascular event. And a large portion of these people at approximately 50, plus or minus 5-10 years, a lot of these people had had a significant major heart attack. And they had noted that this people that had made it to 100, but with a history of a significant heart attack, they noticed that this grouping of people had kind of really bad diet and lifestyle habits up until that heart attack. That heart attack clicked on a light and was like, cool, I got to change stuff. And they made marked changes, right? They didn't make a time machine. They didn't go back and change their history, as cool as that would be. Sci-fi nerds, I'm hoping one day it happens, but they really looked at, okay, I can own what I'm doing today and I can change. And so this group of people sincerely made it until 100 in a happy and healthy way. Did they have to put a lot more energy and effort into their diet and lifestyle? To Dr. Lara's point, being very refined about how and what they were doing, ensuring there was benefit to them. Absolutely. This wasn't a passive thing, but that's something to say that, at least until the point of 50 or 60, I'm sure beyond this, but at least to the point of 50 or 60, you can markedly change and to an extent negate some of the past behaviors. There's always qualifiers to that, but I think that's really important because I think we shouldn't be tethered to past decisions. They might impact how much harder we have to work, but it's not going to per se deny us access to health in the future. Yeah. So Dr. Lara, I'd like to kind of continue Madonna's question, but from someone else. So somewhat similar. So Diane in the chat is saying, she is a beautiful, wonderful 73, but she looks like she could be 55. Congratulations on winning that genetic lottery. She tries to do everything right. So sleep, diet, exercise, supplements, etc. When she had her biologic age run, it was 76. So kind of a little bit older than what she is in spite of looking significantly younger. She has kind of, do you have any insight into that disparity? And, is there anything that can be done to kind of support the turnaround? I would imagine, I'm assuming here, Diane's looking for a younger number there. What are your first, because I think this is a really, really important point.
Oh,
and we talked about this before.
Yeah, absolutely. First of all, I want to put in this qualifier and reiterate the fact that this is not an absolute perfect test, okay? It is a good representation, but you shouldn't put all of your oh, my biological age and, all of it wrapped up in there. If you look great, if you feel great, there is credence in that. So that being put aside, the fact that your biological age was 3 years higher, first of all, within plus or minus 2 to 3 years is actually what they consider average. But there are things, and I would actually have to look at your results and see where there was some disparities, simply because it's not just one number. It's not just your biological age. There's also your cumulative rate, your the subcategories that are taken into account that create your biological age. So I'd have to see, where are you lower? And actually, this works right into what Diane had asked underneath about eating a Mediterranean organic diet. And her age rate shows her diet score at, 18 out of 100, which is, needing attention. Now, the thing is, you're not the first client or the first person I have seen this. I have some clients that are like, but I eat so, well, and what do I do? Well, some of what we have to look at is, how is your gut health? Because you could be eating the perfect, organic, excellent diet, but if you are not absorbing those nutrients, then that's not helpful. You're just, the expression, and this is where we get into nutrigenetics and nutrigenomics. The food that we eat, how our genes metabolize and utilize those nutrients, being nutrigenetics, nutrigenomics being the flip side on those nutrients coming back and affecting genetic expression. There's the epigenetics, okay? So maybe there's something going on. Maybe you have a toxic load of maybe some heavy metals or mold or parasites or something. You have to remember, they eat first, and they can also block the absorption of those beautiful, wonderful, clean nutrients you are putting into your body. Now, I'm not saying that this is the case for you guys, because I don't have your history, I don't have your results, but this is a possible situation that could cause, those types of results in the biological aging, or these little conflicting information.
Yeah. With awareness, there's the beauty of, I think, to your point, and you talked about this at the top of the hour, that this is an evolving test, and we're always adding more and more to the database. And our computers, thankfully, are getting bigger and bigger, and they're able to analyze more and more. And as computing power grows, it's only going to become more and more exacting. Yeah. All right. Dr. Lara, I was wondering, so Dirk has a question here. In brief, he's asking, he's not an academic in this space, so he's not looking for, per se, the textbooks that you studied from, but do you have suggestions of kind of more accessible readings on epigenetics? So, for example, I know you have favorite authors when it comes to breath work. Do you have kind of an accessible author that speaks on epigenetics?
Okay, so yes, Dave Asprey gets into a lot of this. His Smarter, Not Harder. Actually, he's got several series of books. He really dives into epigenetics. I mean, that's his definition in biohacking is and I'm not doing this verbatim. Like manipulating the environment in and around you to affect your gene expression in your favor. I mean, like I said, I've totally, paraphrased the heck out of that, but he does a lot in that space. Dr. Peter Attia, his book Outlive, also reads into a lot of this and things that you can do to expand your life. The research, well, of course, of Dr. Stephen Horvath. He actually is the one who created the Horvath clock, working into DNA methylation and biological aging. There are, you know what? I don't want to use up too much more time for the list. What I will do is I will submit a list of authors, references, and other sites that will be of great interest. I will give it to our magnificent marketing team to add it into the show notes, or wherever they magically put it so people can find it as related to this session. So that way you can have more direct information and access to it.
Beautiful. I like that. I love that we get to share resources here. And thank you, because you're a spectacular resource when it comes to what books and at what different levels, so that people can access this information in the way that they want to. So thank you.
I'm actually writing a note for myself so I don't forget.
You created systems that help. All right. The next question I have here, and Dr. Laura, actually, I don't know. Oh, there's a lot of fanfare in the chat right now, and beautiful ways for this list that you're going to come out with. I like that. I wonder if you can speak to, so when we talk about genes, and this might be a yes or no, but feel free to flesh out the answer a little bit more. When we talk about genes, and the genes that we are very much talking about right now, is we're talking about central DNA. We're not talking central DNA, core DNA. We're not talking about mitochondrial DNA, right? So the DNA we're talking about is kind of, you get one strand from mama, one strand from papa, and there we go. Compared to mitochondrial, where that's just inherited from mama. Do we have any health insights or any current measures of kind of that mama mitochondrial DNA, or is that still something more developing in the science space, especially when it comes to clinical application? Not per se talking about the fun research of it, but are there any tangible mitochondrial DNA methylation investigations or any tools that we can use, or is that a little bit too futuristic for us?
I wouldn't say it's too futuristic. There is research being done. I cannot cite anything off the top of my head right now because it's been a beat since I've dove into that particular research. So that's a fantastic question. And mitochondrial DNA, for all of you, is only passed through the mother, through the female line. But the wonderful thing when it comes to mitochondria, that is our cellular sensor. It is what is reading. It doesn't just make ATP, the energy currency of the cell. It actually senses all the things around the cell and will affect genetic expression in that, say, the cell, that there's something going on and it's like, oh, danger, danger, we need to, explode, implode this cell. It will actually give off inflammatory, markers. It will make the genes express inflammation, or it will have the genes express in such a way to have programmed cell death. Or if it has pressure where, oh, we need to make more of ourselves, it will actually create more, called mitogenesis. So it does so many beautiful things. And yes, there's a lot of research into this. Where we have particular clinical relevance to this works into mitochondrial health. How do we promote health to the mitochondria? How do we make sure that it isn't overburdened? And this works into two particular genes that we have to talk about in our DNA 360 reports and our anti-inflammation, our antioxidation pathway with SOD2 and GPX. These are particularly impactful for the health of the mitochondria of the cell itself. So yes, there are a lot of different things that can be done for the health of the mitochondria. But again, I'm not as specifically aware for mitochondrial DNA itself.
Yeah. I'm not sure if there's validated tools out there, but I think that's an interesting thing that maybe you and I
absolutely.
Now I'm not going to call out one question in particular, but there's quite a few questions right now in our Q&A section that are centering around genes that impact methylation. So, the most famous of these being the MTHFR. And so there's a lot of questions around, what do you talk, like, what do my methylation genes mean in all of this? What does, how do I support these genes? And so I wonder if, we've kind of been talking about DNA methylation a lot. And I think not necessarily that there's confusion, but I guess I can appreciate how some people are hearing epigenetics and all this methylation stuff. And then they're also thinking, but you guys do your methylation panel really, really well. Can you very gently tease out for us, right, genes that have to do with methylation, and then how that is different from, or different and the same, but different and the same when it comes to when we're talking about all this methylation and epigenetic methylation here. Can you tease those two apart for us?
Sure. Okay. Well, first of all, methyl groups are methyl groups. They are set up. So that's not changing. It's just how
And what
they are. It's what?
What is a methyl group for the curious audience out there?
Okay. So when we're talking about methyl groups, we are talking about a chemical, well, it's a molecular structure of one carbon and three hydrogens. So this going back to your chemistry days in high school or in college. So really that's what we call a methyl group. It's when you have one carbon and three little hydrogens sticking off of it. So now this is really important because that beautiful little molecule does some amazing things. It can change one amino acid into another. It can change molecules. It can metabolize and break them down, getting them out of the through our detoxification phases to get them removed out of the body, especially with the toxins. Okay, so it does a wonderful array of things in the body. So teasing out the difference between the methylation pathway, like what we show in our reports. Okay, the methylation pathway, just for all of you out there to give you a little bit of semblance of solidity in what we're talking about. So there are a handful of genes in the methylation pathway that we happen to look at. That's your MTHFR gene, and we actually look at two of them. Your SHMT1 gene, your MTRR, MTR, and FUT2 gene. And there's also the COMT gene, COMT, that plays a huge role because that's actually the protein that puts the methyl group onto the substrate, onto, the metabolite, the hormone metabolite, or the toxin itself, or whatever. So he's the end guy. He's like the LeBron James that actually dunks that ball. She loves the basketball reference. So in those genes, that tells us how the whole process of methylation is working, in that passing of that methylation basketball, from one player to another, in all of these processes, in homocysteine to methionine, and, all the wonderful little details that we're not going to dive into the weeds in tonight about. So now, talking about DNA methylation on our DNA, this is where the methyl groups have to go from the tissues, the cells, inside to the nucleus, where the DNA actually is. And the SHMT1 actually helps to do this. It takes methyl groups from the cytosol of the cell and brings it into the nucleus to where the DNA is. And then it can do its job to be there ready to silence genes, whichever are necessary. And in the absolute brilliance of our bodies and in these cells and these little tiny structures that know what to do and how to do it and when to do it, and they do it so fast, and it's just like we still don't really have a full idea on how all that works. But the intelligence of this, that with other processes, it knows where to put those methyl groups that are now inside the nucleus. So that's where the DNA methylation, but the methylation pathway does all of the other stuff in the body. It handles all of these other processes. So they are interconnected, but they are also doing different functions, different jobs.
There's a bit of self-referencing, right? The very thing that has instructions of how do you methylate, how do you kind of attenuate your DNA is also the very thing that is attenuating those instructions. It's a little bit of a snake that eats its own tail to an extent. Yeah. All right. The next question I think I have here is going to be, so Christy has asked, and I'm going to give a bit of context for this before we jump into the question. Christy has asked, what is the best way to upregulate genes? And she's offered a specific gene, and I'll speak to that because I think it's one that people are often very curious about. But in general, the spirit of the question is, how do you really support, especially the good genes that we have? We talk a lot about how do we quiet some of the less savory genes, but how do we upregulate the good guys that we have? So here she's asking about the P53, P53 gene, and that's looking at tumor suppression, right? So there's a lot of intelligence in the body that says, hey, if we see cancer coming up, we should probably put an end to it. So upregulation of this supports with kind of that natural built-in defense we have against the growth of cancer, right? Cancer really being in a rough, ugly way, saying uncontrolled growth of cells. So she's asking very specifically around, how can we upregulate that? And then I think in general, the larger question of how do we go about upregulating all the good genes that we want? Specific to P350, right? If you look into literature, there's definitely substances. I think green tea is probably the most popular studied in support of upregulation. And I think there is definitely value to specific substances, but I think by and large, and Dr. Lara, hold me honest, I think the literature kind of really boils back down to good diet and lifestyle such that you're preventing I see you're not, I'll let you jump in from there.
Yes, absolutely. And first of all, I want people to know P53, that is considered the guardian of the genome. So, and because it's really important when it comes to its ability to repair DNA damage or destroy cancerous cells. It's really amazing. I did a lot of research study in my PhD in the cell cycle and the different checkpoints that when a cell is getting ready to split into two daughter cells, these checkpoints, very specific. P53 is important in that, and so are many other genes. But in order for those genes to do their jobs, to check the fidelity of the DNA before that split occurs, you have to have the nutrients, you have to have the cofactors. So you're talking about certain vitamins, certain minerals, trace minerals, all of these are very important in the complex that is necessary for this DNA check to happen. And that's really, P53 is just like the main overseer, but there's so many other players that are important players. And in order to have those important players, a lot of them are essential nutrients. Essential meaning we don't make it inside our bodies, or we don't make enough of it. So we have to get it from outside of ourselves through our food, through our water, through the air that we breathe, etc. So this is why it reverts back to what Dr. Krista was saying, that, a lot of this, ways to support good diet, lifestyle, mitigate your toxic exposures, okay? Because everything else downstream kind of works and fixes itself.
And I think a little bit of this takeaway, if we is I think there's always been this search for what is the fount of life, and this hope that maybe there's this one gene that if I look at it and I support it, it's going to fix everything, right? Like we as humanity have been questing for this fountain of youth since time began. And I think we continue in Western medicine. I think a lot, Dr. Lara and I see a lot of people who are like, yeah, I'm super sick, but can you give me just one pill to fix it all? It's kind of that hope that this one tiny thing is going to fix it, right? And we don't want to deny the importance of individual genes and their configuration, but short of certain genes that influence pediatric diseases that you'll find out about pretty quick, most of the balance of the other genes, a lot of what we test for, a lot of this epigenetics really reflects the importance of healthy lifestyle in a way that is congruent with your DNA, right? And so it's the statement of P53, all these things that Dr. Lars talks about, so important, but that unfortunately doesn't replace the need for good food, good sleep, and a lot of laughter, right? Yeah.
That's, in that initial image that I had, shared with everyone, epigenetics is your genes, your lifestyle and environment equals you. Well, your lifestyle environment is the joy, the meditation, the breath, the good food, all of these things. Those things directly impact P53. All of these other, thousands and thousands of genes that we have researched. That's really diving into one specific thing. We need to think of them as leaves. They're leaves on a tree, okay? And yes, those leaves can wither and fall and have problems and disease, but we don't go up to the tree and pluck off the leaf to make it healthy, okay? We don't cut off the branch and go, okay, now we're fixed. No, what you want to do is you want to go down the trunk, you want to look into the roots, and you want to look at the soil. You start changing the soil, start changing that terrain, then you are going to have a healthier tree, healthier roots, healthier trunk, healthier branches, healthier leaves. Everything, it goes right downstream.
Yeah. Yeah. And I think some of the power of knowing your genes or testing, how they are in from a pattern perspective expressing really enables a better what do you do about how do you support those roots, right? Instead of kind of saying, okay, I have to do absolutely everything, it's being wise about that energy and effort, right? And I think that's, when people meet with Dr. Lara, I think that's one of the better things that they take away from their time with you, right? It's not, I must do everything, but here are the things very specific to your DNA, to what's coming up in your health picture. Here's how you focus your energies and efforts.
And I love giving people tools, filling their toolbox with different recommendations, things that they can do, because maybe these are really good recommendations for you, for your DNA, but you can't implement them right now for some reason or another. Well, I would hate for you not to have that information, and, be done with the program and, not have it when you are ready. So not only will I give you, and what we give you, okay, our brilliant, qualified practitioners here in the DNA company, give you the tools to work with and implement right now, as well as some tools to take you and continue on your journey when we are no longer working together directly. So very important. It's teaching you how to fish, not just giving you a fish.
Yeah. I think that's very important here. And I think on that note, I think that might be where we conclude our time together today. We've answered, I think, a majority of questions, at least that are relevant to epigenetics. I do see in the chat quite a few others that touch on nutrition and touch on other things. And I'd really encourage you. We do host a host. That was a little too redundant, but we host a lot of webinars that speak to this, and I'd really encourage you to kind of dig into the archives, because I think some of that has been specifically spoken to. Before though we do jump off Dr. Lara, I'm going to really emphatically say that there's been quite a few requests in both the question section as well as the chat section saying like, hey, please do provide that resource list. And I'm going to say someone even upped the ante a little bit and said like, hey, I'm a medical professional. Could you include some textbooks in there?
I saw that and I made a note for myself to specifically do the nerdy, references to. So
Yeah,
yeah.
do spectacle references as well. So I think we can do that. And I think for those that are really interested, for those that ask some of the mitochondrial stuff, someone in the chat privately messaged us and offered a paper that is speaking to ways to assess mitochondrial health. And so we'll give that a review and then we'll toss it out as well on that reading list so that you all have access to that as well. Yeah. And so I think on that note, that's where we're going to be landing for tonight. Dr. Lara, do you want to speak briefly to an offer that we have for people who are joining this webinar, in particular, a little bit of cost savings should they want to access any of these tests?
Yes, actually, we are giving a special, and I have to apologize. I kind of tuned out a little bit,
It's
but
okay.
our wonderful magical bee
I got you. We're
put
cool.
it in the chat because she is so awesome and has got our backs. So we do have early access for our upcoming promotion for attendees in this event tonight using code SPRING15, that is SPRING15 at checkout for 15% off any 2 or more of the select testing offerings. Those include your DNA 360, DNAging, DNA Gut 360, or PGX. These are non-reimbursable. So the link has been dropped in the chat. I do encourage you all to take advantage. We would love for you to expand your knowledge and even reach out if when you get this testing and you would like to be guided through to learn a little bit more, to work with one of us, we would be delighted to do so.
Yeah. Yeah, I think again, this test by itself is not the be all and end all, but I would also say that of all our testing, I think there's something to be said for being able to view health through many different lenses. Our DNA 360 really speaks to what is your blueprint, and this testing will really speak to how is that expressing, right? And so I think it's I don't think either of us like to over test people, but there is an element of pulling up data to understand how the body is experiencing the world, because as much as we would like to think we're in tune with our bodies, not everything is obvious to us. There's a lot of conditions that are insidious, right? That kind of sneak up on us. And so this is one way in particular, understanding how the DNA is experiencing the world about you and how that's changing the way it expresses. That's very powerful and can lend to a healthcare plan, especially if you're sitting on the healthier side of things and you don't know where to go.
And I do want to mention that with testing can be very important. And me, I'm a data geek. I love the data, but that never takes the place of the subjective feelings you have. You know your body better than anyone. You live in it 24/7. So being in touch with yourself, checking in, knowing your feelings, responses, associating those with thoughts, emotions, really understanding your body and knowing when you do make changes, you can feel or recognize changes that are made, or if there's no changes at all, being in tune with yourself, but never give away your agency of you and how you feel. That is a beautiful metric in and of itself.
Yeah, no, I love that because there has to be harmony between how you want to live and how your DNA may suggest you should be living. We want to live to be happy, not exclusively to be healthy. All right. So on that beautiful note, Dr. Lara, thank you so very much for joining. And while I won't say you consolidated your PhD into an hour and a half, I will say you did a spectacular job humanizing it and offering it in a way that our audience can leverage, but not only the understanding, but how do they apply that? And I think both you and I care about that application. So Thank you so very much for being here this evening. Thank you to everyone in the audience. For those of you I saw in the questions and answers in the chat, some people joined late. We do record these webinars. We'll make sure that's listed out for you. Magical gods of the moderating world will ensure that you guys get that information. And again, we thank you so very much for your time this evening, and we hope we contributed to your growing understanding of health and the sense that you can take your health where it is and continue to improve it. You're not bound to your genes. You're not bound to all your whatever missteps you might have had in your health history. So thank everyone for being here tonight, and we will see you again soon. Au revoir. À bientôt.